What is therapeutic cloning? How does it work? Imagine a future where you can repair damaged tissues or treat serious diseases with your own cells. That future is closer thanks to a technique called therapeutic cloning. It might sound like science fiction, but it is based on real science and has the potential to change medicine. Therapeutic cloning is also known as somatic cell nuclear transfer. It involves taking the nucleus, which contains the DNA from a person's body cells, and inserting it into an egg cell that has had its own nucleus removed. This creates an egg with the person's genetic material. The egg is then stimulated with a small electrical pulse to start dividing, just like a fertilized embryo. It develops into a structure called a blastocyst, which is an early-stage embryo. From this blastocyst, scientists can extract stem cells that are genetically identical to the person. These stem cells are special because they can turn into many different types of cells, like heart, nerve, or muscle cells. Since they match the patient's DNA, the body is less likely to reject them during treatments. This makes therapeutic cloning a promising way to treat diseases like Parkinson's, muscular dystrophy, or diabetes. It also helps scientists study diseases more closely by creating patient-specific cell lines. However, the process is not without challenges. There is a risk that the cloned cells might turn into tumors, which is called tumorigenicity. Sometimes, transferring genetic material can cause epigenetic reprogramming issues, which means the cells might not behave exactly like natural cells. There is also a shortage of egg cells needed for the process, and minor genetic differences between the cloned cells and the original can sometimes trigger immune responses. Ethically, therapeutic cloning raises questions about the moral status of the early-stage embryo created during the process. Some argue that creating and destroying embryos for research might disrespect human dignity. Others believe the potential health benefits justify the process, especially when it can save lives or improve quality of life. The principle of respect for persons urges careful consideration of how embryos are treated, while beneficence asks whether the benefits outweigh the ethical costs. Justice comes into play when discussing who should have access to these treatments and whether resources should be used for this purpose. Laws about therapeutic cloning differ around the world. Some countries ban creating human embryos for research, while others allow it under strict rules. For example, the United Kingdom approved therapeutic cloning for research in the early 2000s, with regulations to ensure ethical oversight. As science advances, researchers are also exploring alternative methods that do not involve creating embryos, which could help address ethical concerns. In the future, therapeutic cloning might become more refined and accessible. It offers hope for treating many serious conditions, but it will continue to be guided by ethical principles that respect human dignity and aim to protect vulnerable individuals. Balancing scientific progress with moral responsibility remains a key part of this exciting field.